So hello guys, this is Kingsuk Panda, your master teacher at Educational Foodies, and I'm your biology teacher. Now you see, today we are going to catch a concept, simple concept of DNA. So particularly I'm writing the heading DNA. So you see DNA, what is DNA? DNA actually, we can just state that DNA is deoxyribonucleic acid. Deoxyribonucleic acid is a very simple concept, and deoxyribonucleic acid is really simple to understand. So we'll go with the term deoxyribonucleic acid. Now DNA, you see, that is so deoxyribonucleic acid. So this thing is particularly distributed into three terms. So deoxyribonucleic and acid. So deoxyribonucleic and acid. So you see deoxyribonucleic and acid, these three terms indicate three different things. One of them is a deoxyribose sugar. Now a deoxyribose sugar and nucleic and acid together may be termed as a or an, I'm really sorry, so an acid of the nucleus. So you see, an acid of the nucleus. So an acid of the nucleus means a deoxyribose sugar containing an acid of the nucleus, or you can just say that it is an acid of the nucleus that is formed of deoxyribose sugars. Now we are going to simplify this study and limit this study to a suppose 15 or 20 minutes thing. So we will go fast and you have to catch up everything in if needed. So you can pause the video just to make sure that you catch up everything. Now deoxyribonucleic acid is divided into three terms: deoxyribonucleic and acid. And if needed, you can just say that it's D, it's N, and it's A are taken to form DNA. So the basic structure of a deoxyribose sugar is given below, and particularly you should know that DNA is subdivided into one term, or you can just simplify the whole thing by saying that DNA is the polymer and its monomer forms DNA. So DNA is a polymer. Now we should know that what is its monomer is. Now DNA's monomer is nucleotide. So suppose it's of n terms. Now n into nucleotides form DNA. N into nucleotides form DNA. That means that nucleotide is the monomer and DNA is the polymer. Done. You can note this down. Pause the video if you like. So next thing what we need is to study the structure of DNA, or just we can study the structure of nucleotide, then form the DNA molecules of it. So nucleotide, its structure is basically formed of three things, basically formed of three things. One is pentose, sugar, and now this pentose sugar is the one we studied earlier. It is the deoxyribo sugar, plus it is also formed of two other things, one being phosphate group, pentose sugar plus phosphate group plus nitrogen base. I've written nitrogen as N2, you have to suppose to write it nitrogen completely. Now these three things form a nucleotide. Now nucleotide, apart from this term, we have another term called nucleoside. Now what is it? So just go with this then. Pentose sugar plus nitrogen base, this forms nucleoside. You see, phosphate sugar, sorry, pentose sugar plus phosphate group and nitrogen base form nucleotide, and these things form nucleoside. Now what is the thing, that nucleoside is a smaller version and nucleotide is a larger version. The difference being there is no phosphate group in nucleoside. Nucleoside plus phosphate group, suppose we term it as N1, suppose just it's for me. So nucleoside N1 plus phosphate group, so say PO4 minus, and it is the universal term. It's just for me, you cannot apply it for examinations. Okay, now nucleoside plus phosphate group is equal to nucleotide, just say it to be 2 N2. Okay, so we've got this, cover it up. Now we have to study the next thing in the course, that is what is the structure of nucleotide. So structure of nucleotide. So basically the structure of nucleotide is very simple, except the hazard being in the deoxy and trioxy case. Now we are just studying the structure of nucleotide, that's for DNA. And you see, in case of nucleotide, we have the very structure of DNA. That means if we study the structure of nucleotide, we will get to know the structure of DNA. So basically nucleotide is composed of three things. So what is this? This is a normal pentose sugar, nothing else. It's just a pentose sugar having 5 carbons. You can see 1, 2, 3, 4, 5. 5 carbons. Nice. Now this is the structure of a pentose sugar. But do not mistake, my guys. Pentose sugar is not the structure of deoxyribo sugar. It is not. Well, deoxyribo sugar is a form of pentose sugar, but pentose sugar is not a deoxyribo sugar. Now, what is the structure of deoxyribo sugar? So basically, I am magnifying the same. Suppose this is the oxygen molecule, and these are the arms of the same. Fine. This is one carbon atom over here, another over here, another over here, 2 over there. Now what do we mean by this is that we will attach an arm over here to just signify that there are bond linkages from over here. but we do not certainly say it as something like just what should I say, hexose sugar, since you can say there are 5 carbons, but it can be another link over here. You can say, but it's not so. It is just supposed to be a single link. Now over here, over this top, this is called carbon 1, this is 2, this is 3, this one is 4, and this one is 5. Now over here we have CH2OH. So CH2OH, this is formed over the C5. There's carbon number 5 atom over here. Now what's found over here? Over here we find hydrogen atom. Over here we find another hydrogen atom. Over here being OH. Over here H. Over here H. H. OH. So this is the basic structure of deoxyribose sugar. This is the structure of deoxyribose sugar. You can just see oxygen atom, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. And over the carbon 1, linkages of H and OH are formed. You see H and OH, then H and H, again H and OH, CH2OH and H. Why this is so? You see, and this was not always in that case. If we just study the sad story of our deoxyribose sugar, then we will find that it was not always so. So we find this term, this annoying term called deoxy. What does it mean? Without oxygen. Now you see, without oxygen, why it is called without oxygen? So this thing is called deoxy since one oxygen is lacking in this thing. So deoxy, it is called so because this is the structure. You see an O has been added over here. Now this is the structure of ribonucleic acid. Ribonucleic acid, you see. So this is the structure of ribonucleic acid. Now ribonucleic acid and deoxyribonucleic acid. You see, or you can just say deoxyribonucleic sugar or ribonucleic sugar. Now we can also mention it as just ribose sugar. You can just mention any term. I'm stating ribonucleic sugar. Now the basic thing is that one oxygen atom being removed from the second arm of the ribonucleic sugar forms the deoxyribonucleic sugar, where it lacks an oxygen atom from the second arm of this deoxyribose sugar. You see, the basic difference between these two are lack of one oxygen atom. It contains an oxygen atom, it lacks an oxygen atom. That's all. You can copy the same. So to move fast, guys, we have certainly not much time. Now we have to study the whole structure of a nucleotide. We have studied the actual main structure, that is the deoxyribose structure. Now we have to move to the main structure of the nucleotide. Now so this I am drawing is the structure of the deoxyribose sugar. So we find this structure at first. It is the base, it is everything. Now, likely over this arm, you see this first one arm, carbon 1 arm. This is the first carbon. Its arm contains the nitrogen base. I hope you have the preliminary ideas of what are nitrogen bases and pentose sugar, and I think you know the basic structure of DNA is. You remember that double helix structure, I guess. So this was the basic structure given by Watson and Crick. And they proposed this double helix structure. Actually, it was proposed by another female scientist. I don't remember the name, but they stole it from her and then published it in their name. But later the truth was discovered, and then special, what should I say, a special address was given to that lady also. But till then she was dead. Anyways, so Watson and Crick proposed this structure, and the base of this thing is that these are the basic backbones, or you can just cancel out the term basic since it might be a bit confusing. Is it basic in nature? No, it's not basic in nature, it's just a backbone, nothing else. You can just remember it as the backbone, and these are the ladders. So what is the ladder and what is the backbone? Backbone is just the nucleotide except the nitrogen base. So this is the thing where nucleotide minus nitrogen base is found, and this is the nitrogen base, and particularly this thing is formed in pairs. So nitrogen base pairs and the nucleotide minus N2. It's all. Over here the nitrogen bases are attached, and over here the phosphate group PO4 minus is attached over here from this arm. Nitrogen base is attached from the first arm to the fifth arm, it is attached, and D, you can consider it as the structure of nucleotide. This is a figure of structure of nucleotide. So it is the basic structure of nucleotide. Now, while we have to study the structure of certainly a whole DNA chain, so we definitely have to convert this monomer into polymers. So to convert this thing into polymer, we have to just attach another nucleotide. But where will be attached? So this is the first arm, this is the second arm, this is the third, this is the fourth one, and this is the fifth. So to the third arm, we attach the next nucleotide. And so on. You see, this is the next nucleotide. So this is the next nucleotide, and this is the next nucleotide, and this is the preliminary or main nucleotide which we started from, or this is the nucleotide of discussion. This is the next nucleotide. You see this arm, this free arm, you can see this is a free arm of the nucleotide, and this is called 5' arm. So why 5 prime? Since it is located in the fifth carbon, and then if just say this arm is free, so it is called the 3' arm. Why so? Because it is located in the third position. You see, third position. That's all. Is the structure of DNA. So this is whole the structure of DNA, and this thing is the structure of nucleotide. I hope you can just note it down. So I hope you studied all these. Now, the next thing is discussing about why about the stages of DNA formation, or where is DNA formed. So DNA is basically formed in the cell, or say in the nucleus, during the interphase. Now during the cell cycle, you see, so in the okay, it's not running. So in the cell cycle we get this thing called interphase. It is nothing except a normal phase, a normal condition of a cell, which is the most active condition actually, but it's called the stable phase or quiet phase. I don't know why, but it is the most active. Most active phase, but it's just called as the stable phase because there is no division. That's all. Now in the interphase there are three phases of the cell. G1, S and G2. Now in the S phase, DNA is synthesized. In the S phase, DNA is synthesized. That's all. And it is synthesized in the nucleus. So you see it's synthesized in the nucleus, and DNA is formed in the S phase of interphase of cell cycle in the nucleus of the cell. That's all. You can just note it down. So next what we have to study is just the importance of DNA, and we will be all done. And I must just say that it's just a what should I say, recap of this chapter. You should and must read the textbook, and this is just a recap. This is shortened version, and this shortened version is meant only for discussion of the basic concepts and the important concepts. And moreover, these are related to the 11 and 9 texts. You should always read the both texts. If you are in class 11, you can just read the class 11 one. If you are in class 9, you should read the class 9 textbook and just go for reference to the 11 textbook if you like, or just watch the video. That's all. Now, importance of DNA. So I'm really sorry for the bad ink. Okay, so importance of DNA. So the importance of DNA is that, first of all, it maintains existence of former characters and carries or transmits genes. Fine. It transmits genes from the parents to the children or the young ones. That's all. Secondly, it maintains the existence of the cell or species of it maintains the existence of a cell itself and also the species of the organisms. Why it maintains the existence of cells? You see, to carry out metabolic functions, we need the order of DNA. If DNA does not order us to maintain to do anything, then proteins of the metabolic things will not be formed. You see, if the proteins are not formed, that means the metabolic activities are also not done. Right? So these two are the main characteristics, and the third being, and I must mention over here, from parent to children. So from parent to child, it maintains the existence. And the third one and the last one importance is that it helps us to study the variations among the organisms, or it helps in variation. That's all. You can also just simplify the whole thing by saying that DNA helps in distinguishing organisms. Distinguished or distinguishing characteristics of organisms. Fine. So these are the three main importances. I hope you enjoyed the class and enjoyed this topic. We are ended over now, and I think this is the last thing of the discussion. Thank you very much. I hope you like the video. Share the video, comment on the video, and just tell your friends to subscribe. You also will subscribe to the channel for more daily updates. And if you like my face, just click on the comment section, click on the subscribe, and don't forget to click the bell section. Just wait for a minute. Go to our Facebook ID. If it's not given in the link, we are really sorry. We will provide it later. Go to our Facebook ID. Just like our channel and like our Facebook page. I hope you like the videos, and for upcoming comments, don't forget to click that bell icon. Fine. And this was Kingsuk Panda from Education Foodies. Thanks.